All right, welcome everyone, and my apologies for the delay in getting this webinar started. First and foremost, I want to thank everyone for attending today, and specifically our speaker, whom I will introduce in just a minute. This is the next installment in our series hosted by Sino-Biological of webinars called BioTalk Tuesday, where we speak with leaders in their respective fields about cutting-edge technologies in life sciences. So in just a second, I'm going to introduce our esteemed speaker. Before I do, though, I just want to mention very quickly one housekeeping item. I ask each individual to please, if you have a good question, ask it in the chat box. And at the end of the seminar and lecture, I will run through the questions and verbally ask them of Dr. Kittle to try to get as many of those questions answered as possible. Please also feel free to just shout out in the chat box to everyone and say hi and let us know what part of the world you are from. So with that, I will now introduce our speaker, which is Dr. Joseph Kittle. Dr. Kittle is the Chief Scientific Officer of CytoSPAR. He'll tell you a lot more about his company in just a minute. Dr. Kittle graduated with a BS in chemistry from Ohio University and received his doctorate in chemistry from Harvard. He also completed his postdoc at The Ohio State University in molecular genetics, and he's held positions at Battelle, as well as an assistant professor in the Department of Chemistry at Ohio University. Dr. Kittle has over 30 years of experience in biotechnology product development. And the title of Dr. Kittle's talk today, as you can see, is Harnessing the Power of T-cells Using a Programmable Receptor, a Practical Approach to Diagnostic and Therapeutic Applications. And so with that, Dr. Kittle, I welcome you, and I will stop sharing my screen so that you can share yours and start your lecture.
Thank you. Thank you, Rob. And thank you, everyone, for joining in today. What I want to tell you about is some work that I've done with a company called CytoSPAR. And in some ways, it's a culmination of years of sort of a progress through and across molecular biology. You know, I started out in chemistry and got pulled over to the dark side of biology early in grad school. And I got involved with RNA and DNA work then. I went from there to being able to do a lot of genomic work at Lark Technologies in Texas. And really along those ways, we were just trying to figure out how things work and how to use technology to understand biology better. From that, I progressed into synthetic biology because my interest in RNA molecules and their structure naturally led to the idea that we would like to be able to control these structures and the proteins made from them. And so around the year 2000, I got really interested in making synthetic genes and started a synthetic gene company at that time. That field progressed to the point where really the most interesting thing wasn't just making DNA, it was being able to design things. You know, if you could make any sequence, what would you make? And I think the answer came out to be, for a while, let's make some proteins and we can design really interesting proteins. And now I think where the field is really moving toward is the idea that now you can control biology, and the natural area to control biology is really at the level of cells. And so what I'm going to show you is how we were able to harness the power of T cells, which are nature's control mechanism for all of your immune system. And be able to control that in a way that we could develop new diagnostics and even potentially new therapeutics based on the activity of these very powerful policemen of the body. So I'm going to show you a little bit about T cells, and I'm going to show you some examples of how we were able to modify the T cells so that we could control them. So on this first slide, I want to talk a little bit about engineered T cell receptors. So I'm going to cut to the chase, and then I'm going to come back and explain what we did. But what we were able to do was to engineer T cells so that we could target them and control their activity so that they went after specific targets that we wanted to activate on. So when a T cell activates, it does lots of things, and I'll show you that in a moment. But in order to get them to activate, the activation goes through something called the TCR, the T cell complex. Now, T cell complex, like a lot of membrane complexes, has got a lot of components to it, and all of them are important. And so if you look at this engineered SPAR cell, now what's a SPAR cell? A SPAR cell is really just a T cell that's got a surface programmable activation receptor. Let me show you that in the title slide here. So surface programmable activation receptor. So we designed a version of the T cell receptor that we can control. So if you look at how these, let me see if I can get to the full screen here. If you look at the way the T cell receptor works, it's this complex that's got an outside part and an inside part. And when the outside part comes in contact with a target, these T cells are able to target cancer cells or viruses or bacteria. Then it activates what's on the inside, these little domains here called ITAMs. This is T cells do a lot of things in the body. They turn on B cells, they kill cancer cells directly. And they're just the center of the immune system. So naturally, over the years that people have worked with these T cells and come to understand their power, they've made versions of this T cell receptor called chimeric antigen receptors. And what they did was they took different parts of this T cell receptor and fused them together into individual single proteins called chimeric antigen receptors. And these were able to control the T cell. And in order to be able to target something, they would affix on the outside a binding domain that would bind a specific target, say a target like CD19 on a cancer cell, or maybe a target on a virally infected cell. And so you could activate the T cell using a single protein instead of this big complex. But there are problems with that. And the problems are that the T cell is a very subtle mechanism, and it can turn things both on and off and control its own activity. So that's kind of lost in these CAR T cells and their CAR T cell receptor. In addition, being able to remake these is complicated because you have to design an entire new complex protein. It's a membrane protein, and that's not easy to do. So one idea was to be able to make these cells with an antibody on the outside that would bind other antibodies, which is not a bad idea. But again, it's a chimeric antigen receptor. And so being able to control this in the body is kind of hard to do. So in our case, we looked at this in a different way, and we come at it from a different point of view. We weren't trying to do CAR T therapy when we started doing this work. We wanted to turn the power of the T cell in order to be able to create diagnostics. Now, our founder, Charles McBrady, worked for Abbott for many years, and he understood that better diagnostics were needed. And so he came up with this idea of doing T cells as a diagnostic. And what we were able to do in the lab was to create a version of the T cell receptor, which was programmable. Now, how do you do that? Well, we took a component of the T cell receptor, and instead of glumming all these pieces together, we just simply put a domain on it. There's something called, you can barely see it here, FcγRI, which is a binding domain that binds antibodies. And we picked one from mice so that we bind specific mice antibodies. And we fused just the outside of that to the outside of our T cell receptor. Now we have a means to bind mouse antibodies on the surface of a T cell receptor. Now, normally this part of the T cell receptor is what determines the specificity of the T cell receptor. And what we did, we switched it so that the antibodies we bind here actually determine the specificity of this T cell center. So this T cell receptor goes after the thing that we add to it. So we grow these cells up and we can add antibodies, and now we can control the activity of these T cells. So we thought that was pretty cool, but we felt like it was a little limiting to be only able to bind antibodies. So we made several other versions of this, including one where instead of we attached something called monostreptavidin, and in fact enhanced monostreptavidin to the outside of the T-cell receptor. And we found and patented, curiously, that you could actually attach this thing to other parts of the T-cell receptor and get the same effect. That is, we can now control this T-cell receptor by use of this tag on the outside. And I'll show you how that works in a minute. But what's interesting about that is now we have the ability to make T-cell receptors that can bind using the streptavidin domain to biotinylated proteins, or anything biotinylated oligonucleotides or carbohydrates, or anything with a biotin. We've now got it set up so that that will bind to the surface of a TCR. Now, why does it work here on this protein, or this one works over here on this protein? And that's because you need to understand the way these TCRs work. The way they work is that when they bind something, and then several of them bind to the same kind of target, then they cluster together. And that clustering together is what triggers off the T-cell activity. And that clustering is really important because it does two things. One, it enhances the overall binding, but it also makes it more specific. So if you don't have multiple binding events, then the T-cell won't fire off. So you get a lot less false positives, and you get very few false negatives because it's a very sensitive system. So how do we use this? Well, we create something I'm going to talk about in a moment called the targeting TDM. This is the targeting molecule that binds to the this EMA domain of the TCR we engineered. So basically, it's going to have a biotin down here that will allow it to bind to the TCR. So let me show you a little bit more about how we did this. It's pretty straightforward. We made gene constructs where we fused the FcγRI directly to CD3 zeta. That is this example. And then in the other example, we fused the monostreptavidin to CD3 epsilon. We've also done it with other TCR proteins, and that's all patented and work. But what we were able to do then was to make different T-cell lines, one with this kind of receptor and one with this kind of receptor. So we have different options. In addition, we did something that makes this into a diagnostic. That is, we put a reporter protein in there. This AEQ stands for something called Aequorin. It's a protein from jellyfish that makes them flash. So we borrowed from nature not only the cells, but we also borrowed a jellyfish gene that when these cells get excited, they start lighting up like a jellyfish. Very convenient when you want to detect when they're excited. So we make this protein inside the cell using this constitutive promoter so that it's on all the time. This protein's continuously there, but they're not flashing. The reason they don't flash is because in order to flash, they need calcium. So let me tell you a little bit about that. So T-cell biology, it's very interesting stuff. It's pretty complex, and it's been studied a lot. And the reason why it's complex is it has a very important job in the body. It has to detect cancer cells and infected cells with viruses or bacteria without killing the body by attacking your own tissues. So when there's a lot of steps in T-cell activation, but the main one, as I said, was the TCR binding to a target, clustering, and then firing off. And what happens inside the cell is that you go through a cascade, amplifies the signal. So just a few little TCR events on the surface causes the entire giant cell to activate. And it makes phosphorylated proteins, and then that phosphorylates some other molecules. And then eventually you release calcium through some ion channels in internal stores of calcium inside the cell. So what's going to happen is when this TCR fires off, a rapid cascade of amplification events occur, and then calcium ends up being released from some calcium stores inside the cell. Now, eventually, this signal propagates to the rest of the cell and starts turning on cell proliferation and cytokine production and all the attack modes that the cell uses to kill the targets. But that takes time, and I'll show you that in a moment. but immediately you get this calcium release. So why did we put Aequorin inside the cell? It's because Aequorin will sit there and be inactive. It will bind a molecule called coenterazine, and coenterazine, when it's cleaved, will give off a bolt of blue light, 462 nanometers. So it's a very bright amount of light that comes out of the cell when this Aequorin fires off. So we sit there with our T cells loaded with coenterazine on the Aequorin, no calcium. And then when the TCR fires off, because it sees a target, calcium is released, and that activates the Aequorin. Now this thing goes from completely off to completely on over a fairly narrow range of calcium. And the trick is that it uses cooperative binding. So you have not just one calcium ion binds, but several bind in cooperation. So if you understand the kinetics of cooperation, what happens is that goes from completely off to completely on in a very distinct switch. So we switch on the Aequorin and get a flash of light, which we can detect. So that's sort of the basis of our diagnostic, a controllable TCR that goes after targets we want, and then inside the cell, a bolt of Aequorin that gives off light when it does that. So I showed you a little bit, a complex diagram about all the things that happen inside the cell. And that's sort of temporally in all the actors. Here's something that talks about the time frame, the temporal domain. So within seconds of TCR triggering, you go through that entire cascade of proteins ending up with calcium release. And then within hours, you get the other domain, other things turn on. But that very quick signal is what we're able to take advantage of. Let me show you how quick. Here's an example of where we've taken our T cells that are loaded with Aequorin and coenterazine, and they're sitting there with no activity. Then we add in an antibody, anti-CD3 epsilon, which will trigger off the T cells. So when that happens, you get a large release of light. Now we're measuring over here RLU, relative light units, and they're millions. It's in the millions. The baseline down here is 1,000 or so. And we're going up to a count of several million just by triggering off the Aequorin inside that cell. So if you don't trigger it, if you use a different antibody, you get no activity. If you add an antibody specific to triggering off the T cell receptor, the well-known anti-CD3 antibody, then you get this burst of light. When we saw that, we knew that we had the ability to monitor the T cell activation event. And look at the time frame. Here we are at around 1 minute, and already you're starting to get a huge burst of light. So in about 2 minutes, you peak out in this particular assay at over 1.5 million RLU. So this is a very powerful activation event, and we can monitor it. And you monitor it in the standard laboratory luminometer in this case. So we wanted to be able to use this as a diagnostic, but it's important to know that diagnostics labs need to have easy to use reagents. So we figured they are not going to grow these cells up fresh and use them in the lab. Also, we wanted to be able to do quality control on these. So what we figured out was we could grow the cells up, get them ready to use, and then freeze them. And when you thaw them out, then they need to be active. And I'm showing you on this slide that we can actually do this. Now I'm going to show you an assay here where instead of triggering off TCR with a TCR specific antibody, we actually used an antibody to E. coli LPS, that is the lipopolysaccharide that covers the surface of E. coli. And that's a lot, by targeting that, we can identify E. coli in solution. So we use an antibody against LPS, and this antibody binds, it's a mouse antibody, and it binds to our modified T cell receptor, the one with the FcγRI I told you about earlier. So when you mix these together, the antibody and the cells, not much happens. Then when you add the LPS, you get a huge signal. And here it is with fresh cells, and you get thousands of, 40,000 light units. The control here is where we use we use another strain of E. coli, E. coli 0157, lipopolysaccharides a little different, doesn't react with our antibody. So the antibody positive control with the right target gives a big signal. Antibody with the wrong target does not. And then we showed that this same thing takes place in frozen cells. And again, so we freeze the cells, thaw them out, and they're ready to use. So basically we have a laboratory reagent that we can provide that when mixed with antibody becomes very specific as a diagnostic. So here it is for E. coli, going after E. coli 0111. And again, the time frame, here we are at 61 seconds, and you've already got a measurable signal. So we went through and developed a lot of these assays against lymphoma cells, and I'll show you that one in a minute, using a biotinylated antibody as our target detection molecule at TDM. So this is where we used a biotinylated molecule as our TDM. We've also gone after human melanoma cells, mouse melanoma cells, and I'm going to talk about some of the work we've done with MERS and SARS and influenza and bacteria. So there's a whole lot of these that we've been able to put together, and we've been able to do it with both of our systems. So they both work. Either you can use a biotinylated target detection molecule or a mouse antibody or something fused to a mouse antibody, as I'll show you. So we also were able to use this not just to detect purified LPS, but here's a case where we use a luminometer that we built to detect a very low concentration of E. coli. So 10,000 E. coli is about the smallest amount of E. coli you need to detect a urinary tract infection, for example. So 10,000 CFU of E. coli going after E. coli 0111 again, only this time instead of purified LPS, it's the whole E. coli. You get a very strong signal, 30,000 RLUs, again, the very rapid time frame. And if we use a control culture of E. coli, another isolate, E. coli 0157, you get no signal. So we're able to use this in a diagnostic mode to detect E. coli in environmental samples or potentially in human patient samples. So here we see detection of a virus. So here we're looking at the influenza virus. And I don't know if you can see that on your screen. We've got the screen sharing up here. But in this case, we've got influenza going after the H1N1 protein. See if I can move this out of here. There we go. Influenza going after the H1N1 protein or a different protein. And we're using a mouse anti-H1N1. And so you can see that we can detect viral protein this way. And again, the time frame is pretty quick. So here's an interesting one. This is the world before it was changed, before COVID-19. And we were interested in a sort of an important virus called MERS. It's a coronavirus, which were kind of mysterious at the time. And it's in Middle Eastern camels and sometimes gets into humans and kills them quite ruthlessly. And we were able to develop a test against the spike protein. Here's purified spike protein. It didn't take us long to do this. We bought purified spike protein. We got the antibodies. We were able to show that our assay would detect the purified spike protein and not detect the SARS protein. Now, SARS is another coronavirus, which was a real problem in a few years ago. This is the original SARS, and actually had some outbreaks around the world. And we were able to distinguish between MERS and SARS in the same test. Now, these are with purified proteins. So we thought, what about actual samples from animals? At that time, there weren't any coronavirus human samples available to us. So we worked with a collaborator at Colorado State University where we got nasal swabs from alpaca that were being tested with MERS. So MERS infects camels, alpacas in the camel family. So they have big nostrils, turns out. So we were able to get nasal swabs and show that we could detect in 2 different animals. This is one animal, animal 7, which detected, here's day 3 sample after infection, here's the pre-infection sample. And so we were able to detect a clear difference between animals that were infected and uninfected in these alpaca. Now, this was back about 2 years before 2019, about 2016, 2017 timeframe. So we had a coronavirus assay available to us at that time, not knowing what the future was going to hold. So there's a viral test here. So we've done virus, we've done bacteria. Let me show you something else we've done with the test. In this case, we're flipping the test around. Instead of trying to detect a target with an antibody, we were trying to detect an antigen with an antibody. We're detecting here an antibody using a surface antigen. So here we've taken a protein, happens to be tumor necrosis factor, that's been biotinylated. So this biotinylated TNF was available. We bought it, and we were able to use it with our biotinylated SPAR cells. So remember, what we're doing is we're taking biotinylated TNF and putting it on the surface of our T cells on the receptor, on the TCR. So anything that would cluster that TNF would cause the TCR then to fire off. Now, so then we took a antibody to TNF and put it into solution. And it didn't bind to the T cells. What it did, it bound to the TNF, and it caused the TNF to come together, and then that caused the TCRs to cluster and gave us a strong signal. So you can see here again, we get that rapid signal increase compared to, and here we were showing that we were getting a signal on the T cell receptor. Now we used a little bit different kind of control here, because I always like to have, you have to have good controls in your experiment. Since this is a biotinylated TNF, we simply added a batch of biotin to the solution and showed that biotin would block this signal. So this is the same setup. The only thing is we put in biotin into the solution, and we're able to show that in the presence of excess small molecule biotin, it blocked the ability of the target detection molecule to bind to the surface. And so even though TNF was being attacked by the antibody, it wasn't resulting in a signal. So that's the kind of controls we like to run to make sure that everything makes sense here. After all, TNF is a biomolecule. So let's talk a little bit about target detection molecules. And I'm trying to see, maybe I can move this out of the way here. There we go. Here we have part of that diagram I showed you before. Here's the extended enhanced monostreptavidin that's down here bound to the TCR. But I wanted to focus on TDMs. And one of the reasons I wanted to focus on it is because Biologics has helped us making some of these TDMs. This is not a commercial. I just want you guys to know that we appreciate the work you've done for us. In this particular TDM model here shows that we've got on this end a homing tag, which is a little bit of protein, a domain that can be specifically bound with biotin. And this is what's going to connect to the cells and help arm the cell. So this is the homing tag. Then the rest of the target detection molecule, the TDM, has a stabilizing core region. Now this is just, in the world of engineering proteins, you need to have a stable structure to build off of. So we put a domain on this end, and we put a domain on this end, and in the middle we've got this stable piece of protein that is just used to help us purify the protein. Very easy to work with. And then on the other end, we typically clone a binding domain. Now this binding domain may be the variable regions from a antibody. It may be some other molecule that has some interaction with a target, or in fact, it may be an antigen, like I just showed you, So we can actually put on this, instead of putting an antibody domain, we can put the target of an antibody. So you can see by designing these different TDMs, we can dial in the activity of these T-cells that we've engineered. And that has a real practical aspect to it. So we can grow up big vats of these T-cells and just have them on hold or have them frozen away, waiting for us to develop whatever targeting system we want, and then test those and go out to use them in the field. So what if you make this targeting domain, this target interaction domain here on the end, what if you make it the SARS-CoV-2 spike protein S1? So what we're doing is basically putting this antigen target on the end of our target detection molecule. And in fact, we built around this what we call the serology assay, where we were able to take biosensors that will bind TDMs. And in this case, we made a fusion protein between the spike protein and IgM and the mouse IgG2A constant region. So the TDM is IgG2A constant region and SPAR and spike S1. Now that's pretty reasonably easy to produce. And then we put that in solution with our SAR, with our cytospar cells that are able to bind these mouse antibodies. And so now we have it targeted. Now, what do we use that for? Well, if you take human patient samples and you want to know whether or not they have antibodies to S1 spike protein, you simply have to mix it with these cells. Now, our cells don't require that the sample be purified or treated. It can take raw serum. In fact, these T cells work with just about any body fluid, because T cells normally work in urine, in the serum of the body, in cerebral spinal fluid, even in saliva. So there's no enrichment and no purification of biomolecules that are presented to these T cells. That's a huge advantage in the clinical world. So we take our ARM T cells, mix in the patient sample, and you get, if you have the case, if the patient has antibodies to this spike protein, then you're going to get a large signal out. And you can control that against negatives where the patient doesn't have these antibodies. And in fact, if you want to change the specificity, then you can look at different spike proteins, because the spike proteins have all been sequenced and can be, you can make target detection molecules out of them. So we wanted to move this a little closer to the clinic to see how this works. So we started to look at different concentrations of antibody to determine the sensitivity, and the thing's pretty sensitive. With this particular setup, we were able to sort of look at, high levels of antibody, 6 micrograms per mil, all the way down to less than a microgram per mil. So we were able to do the statistics and come up with some things that you typically see in diagnostics, like limit of quantitation, or LOQ. So this is a semi-quantitative assay. The amount of light integrated under this curve. And then the limit of detection, what's the smallest amount you can reliably detect? And then what is the limit of blank? So this is working toward those type of regulatory questions you need for a full-blown diagnostic. And then we actually went and got clinical samples. So you can get authentic negative samples simply by going back to patient serum that was available from patients where it was collected before the pandemic. So we have we got 20 negative samples and we got 20 positive samples. Here's a few of them on this graph here. You can see the high antibody concentrations give you these blue lines, and some lower concentrations give these green lines. So down in this part, and this scale is deliberately showing how wide a variation you see in patient samples. But you can sort of look down here, and the math will allow you to pull out these different samples. And in fact, from 20 positives, expected positives, which were tested positive, people who had authentically experienced COVID, 19 of them were positive in our tests, and 1 of them was negative. So I don't know if that's a false negative or the person just didn't acquire antibodies. We're learning that happens. And then in the samples collected before the pandemic, we got 20 of those, and 19 of them tested negative, and one of them was a weak false positive. So this has a concordance of around 95%. In a larger set of studies that we did with authentic patient samples, we were able to see somewhere between 96 and 98% of the expected results, both in the false positive and false negative. So this is a pretty good test, and it's semi-quantitative. So to stop here for a moment, I've got about another 8 minutes before I stop and let you ask questions. But just to look at these, the advantages of our system are ready-to-use components and this minimal sample processing or enrichment. Remember, these T-cells work in biological samples or physiological buffers. So we don't need to, and the sensitivity allows us to do a lot of targets directly from blood or serum or saliva. It's very rapid, and it's rapid in two ways. First of all, the assay time, as I showed you, is rapid. In fact, one of the things you have to do when designing these assays is make sure that you read them fast, because they read really quickly. So, and then also the time to development. As fast as we can either purchase or design and create these target detection molecules, that's how long it takes for us to kind of get this development process underway. And using our pre-made cells, we're able to quickly get to useful assays. And in fact, one of the reasons we work with Sino is to be able to make these target detection molecules quickly, sort of, here's what we need, and you guys send it to us. And that cuts our time to development and also development and cost. So the reason you're in business actually helps us a lot. So that's why I wanted to do this seminar. It's very sensitive, as I showed you, which is a key, because then you don't have to enrich things. And it's also very reproducible. You notice in all those graphs I was showing you, there were multiple lines. And so we can reproduce this over time on the same day or over different days, and then even in different labs. This test has now been used in multiple labs in Florida, Colorado, and different places around the world. So sort of the last thing I wanted to talk about was our work in cancer biomarkers. So remember what T cells do for a living. The reason you have them is that they go around your body and they interrogate cells, and they will interrogate a cancer cell by looking at the surface. Now, with our high affinity target detection molecules, we don't need the entire MCH complex in order to trigger off our T cells. But what our T cells are able to do are able to interrogate living cells. So you don't have to blow the cells up to do an ELISA or do some sort of PCR test with purified components. We just mix our cells in with whatever the sample cells are, and they can read the surface directly, just like T cells normally do. So here's where we're looking at Raji cells. Now, Raji cells are B-cell lymphomas, and they make CD19 on the surface. And we used a biotinylated anti-CD19 antibody. And here you're able to see that if you mix these human lymphoma cells with our T cells, our engineered T cells, you get a pretty strong signal here. This is 40,000 RLU compared to a baseline where we also mix them with cells, but in this case, we mix them with the cell line K5262, which doesn't have the CD19 marker. Curiously, you notice the time frame's a little different. We've been talking about these. Normally, the peak would occur around here, around 60 seconds starting, and then go off and be done in about 2 minutes. Here it takes longer, and I think it's because when a T cell is interrogating another cell, the targets on that cell have to raft around in the lipid bilayer and come together so that the TCR can cluster. As you can see, we get a long signal. I mean, we did this with other markers. Here it is with human melanoma, looking at the CD133 marker, and we're using a mouse anti-CD133 antibody. And so we're using a Glomax here as commercial luminometer. And again, you see this longer time frame, but very strong signal. And in this case, we're looking around 10 to the 6 cells compared to, again, our control cells. And you can also, typically in these, just for other controls we typically run, we'll do it without the antibodies and some other tests that we do. And here it is with a mouse melanoma. So you're getting the picture here. We can develop assays pretty quickly for these different cancer cell markers. Here it is. If you look at the different commercial antibodies out there, not all of them work. Not a surprise there. Not all the antibodies work the way you think they do. And some of them work really well for TDM. So out of about 10 antibodies we tested across 3 different biomarkers, about half of them work, about a third of them work. So, and they work well when they do. So we thought, okay, so we can detect purified cells in culture. What about in blood? Now, we didn't have access to cancer patient cells just yet. We didn't want to go that route. So we made what are called contrived samples, where we mixed Raji cells in with human blood. And we were able to detect a small amount. Here's 10 to the 5th Raji cells in blood compared to 10 to the 5th added culture cells that don't create CD19. And you can see a pretty good signal difference here. If you increase the number of cells, where we're using 10 to the 7th cells here, using a biotinylated anti-CD19 as our target detection molecule, you get a larger signal. So the signal is proportional to the number of cells and to the type of marker used. So I'm going to stop here in a minute. I just wanted to point out, though, that in our TCRs, here's one with the EMA. Remember how that works. They actually are in a what's called a Jurkat cell line, and they're very similar to just regular T cells that you would have in your body. And so other people have done TCR modifications where they've tried to go in and engineer the TCR with different TCR domains. That can be kind of hard to do because the TCR, this part of the receptor is pretty finicky. Or you can make these chimeric antigen receptors, but then they're hard to turn off and turn on, and so they're a little bit hard to control. Oh, I wanted to go back to well, we'll go forward to this. Anyway, so one of the things we wanted to do was to look at whether or not our T cell receptors were actually causing the T cells to fully activate. Was it just calcium, or were they actually doing some of the things that wild type T cells do? And so we looked at IL-2 release. Now, when T cells get excited, they release cytokines, and one of the most famous ones is IL-2. So this is an easy assay to do. We mixed the cells with just the cells. You don't get any IL-2 release. You add the cells with PBS or cells with our target. In this case, we're using the LPS system I told you about at first. So, and then if we add the cells in our anti-LPS antibody, you don't get much activity. But if you add the cells plus the antibody plus the LPS, you get this large increase in the amount of IL-2 release. So the SPAR cells we create are actually real T cells. I mean, they still act like T cells. They release IL-2, and they actually upregulate. I'm not going to go into the data here because of the time, but they actually upregulate surface markers that show T cell activation. It takes hours for this to happen, as opposed to the seconds it takes for our calcium release assays. But again, we can see T cell activation, and we were actually even able to show that these T cells were able to kill cultured Raji cells that are human lymphoma cells in our experiments, where you add all the right components. You add the antibody and you add the T cell, our engineered T cells, and then you add these Raji targets, and it kills them. So I'm going to go back. I'm going to stop there and just kind of leave it on this summary slide here and answer questions.
Thank you, Dr. Kittle, for that excellent presentation. We sure appreciate it. Fascinating technology based upon T-cell based cell culture for diagnostics. So thank you again. And we've got good questions from the attendees today. I'll just get right into those, starting with a question from Kabita Sharma. Specifically, Kabita asks, homing tag indicates housekeeping genes are being immunofluorescently labeled. I believe that's a question. Do you understand that?
Yeah, so what we're trying to do is design, the homing tag just means that a way of, it's a made up term. It's not a term of art. So I'm just calling it a homing tag. So on the end of the target detection molecule, on one end, we want a piece that can tie onto our engineered T cells. And that homing tag could be a biotinylated domain, just a biotin hanging off of a protein, or it can be a mouse constant region from an antibody. But that tag just means that it comes back to the T cell. And so when you first add the TDM to the cells, they mix, and then those things bind to the surface of the T cell. That arms the T cell. And now you add the sample, and it's got whatever your target is, and the other end of the TDM then gets active. I hope that answers your question.
Thank you. The next question is from Florin Zugan. Florin asks, is TCR-mediated endocytosis limiting the glowing time extent?
That's a great question. So there's a couple of things that are probably limiting. One, we run out of selenterazine, so the enzyme doesn't really turn over very fast. The other thing is, I don't know. I would assume yes. I would assume that endocytosis is something that's going to have a major play, especially if we're using the cells in other modes beyond diagnostics. I don't think endocytosis occurs quickly enough for that 60-second response we see. But after that, I think it might. And also just, we open up other calcium stores and external calcium stores. So it gets complicated. And we were kind of relieved to see that even though the biology is complicated, the signal we see is usually pretty straightforward. But I do think it's limited by things like that.
Great. Lauren also suggested that a solid phase capture of the ligand design might confer advantages, both in involving co-receptors and allowing maybe multiple target detection. Do you have an opinion on that?
Yeah, I think this is just early days with this technology in the ability to capture, to change the way the system works or to enhance the system, add other components. I will say that we've done some experiments I've not talked about here using multiple components of protein in solution. And you get some really intriguing tests. Like, for example, we developed a neutralizing antibody test that way. So she's exactly right. Using
good
solid
suggestion
could really create a whole new way to work with these cells.
Yeah, maybe a good suggestion by Florin y'all want to test out at some point.
Yeah.
Rita has another question, looks like a good one. Do the relative light unit values hold relevance only in terms of quantifying the T cell enrichment as per fluorescent tags added, or do they indicate its specificity and functionality as well? It's quite a question.
I think what we're doing now is really a very simple sensor. All we're doing is just having a photo eye look at the sample, right? And it says how much light's coming out. You could also use microscopy or some sort of more functional. We were able to also do this in multiple colors. I won't get into that here. But yes, you can definitely, a CCD camera, for example, might be a good way to address this and start looking at image analysis as well. We aren't doing that yet.
The CCD camera is an old-fashioned technology, but certainly extremely sensitive. So
yeah,
might be a good option to test. And Sang-Hoon Kim asks, does density of the T cell receptors affect the sensitivity?
Yes. And And that's one of the things we have to control with during manufacture. I haven't talked about, the challenges of manufacturing an actual product out of all this. But being able to control that, we actually looked at lots of different, there's a lot of work going into making this the first time. And so getting the right amount of receptor on the surface and having it intact took a lot of work, getting the right promoters and the right construction. That's part of our patent portfolio as well. So, so yes, the amount of receptor is really important. And so that we've done some work in that area.
Yeah, and Sangu asks, is there an optimal number, or do you know how many T cells, or actually the T cells, are needed to see a reasonable signal? Do you have to have a certain density there?
Yeah. So we, in our assays, we varied the number of T cells. And again, it's work I haven't shown, but we don't need a huge number of T cells to see them. In order to get the maximum sensitivity, we use about 1 million T cells or more, but we can actually detect them with smaller numbers. It just becomes less sensitive. But yeah, the number of T cells is really important, and it's part of the optimization process.
Skylar Chuang asks, can you do this with other types of immune cells like macrophages or DCs?
We have done it with other cells. Again, part of the work we've put out there, certainly with B cells and some others, and also with other receptors besides the TCR. It's just TCR is really good. Some of the others work as well. We've done some work with B cells and some others, but I think that's an opportunity to look at, and these other cells have different functions within the immune system. So you could potentially control other functions in the immune system.
Sounds great. So just back to Skylar, another really good question, just a general question. Can you mention some of the hurdles that you're currently trying to overcome with your technology?
Yeah, that's a great question. So one thing is to streamline the production of target detection molecules so that we can have a soup to nuts approach. And using better modeling techniques and that. Another area is just manufacture. Cell manufacturing is a challenge. Most places that do a lot of cell work are actually just making proteins, and so they're working with CHO cells, or they're working with T cells, but they're working with human T cells. So working on the manufacturing part of this, being able to, making the proteins is actually the easy part. The harder part is growing the cells up. We've got that, we've made lots and lots of batches of this and froze them down. And so we can do that, but being able to scale it to the huge amounts you'd need for millions of assays is really just one of the things we're working on right now. That's a big challenge.
Sounds great. Well, we're at the bottom of the hour. There were a few other questions. I apologize, I wasn't able to get to those. I would ask if y'all can just email those over to Sino. We can get those answered for you by Dr. Kittle.
Yeah.
Kittle, first and foremost, let me thank you for an excellent presentation. We also appreciate the kudos that you gave to Sino throughout your talk. It's always nice to hear. And I'm glad you're very pleased with our antibodies.
Yeah.
Indeed, a very fascinating technology that you have there. And I think I congratulate you and the other team at CytoSpar on your accomplishments. And I know that y'all have a bright future.
Yeah, and I have our team is really the key to what we've done. Joel's on the line here. He's my standby. And just a group of people that are remarkable. Some of them we pulled into the company from undergraduates, others are PhDs, and they have worked so hard. And I'll tell you, I've not worked with better people in my life. So including all my experiences. So I'm really proud of the team. And our CEO, actually, believe it or not, cares about the technology. Imagine that, and dives into it. So it's a great team we have working with. So I'm privileged to do this, I'll tell you.
Congrats to all these folks on this acknowledgement slide. And Joel, I know you're attending. Congrats to you as well. And thanks to the attendees. We had great turnout today. I just have to mention, we had folks from Zimbabwe, Ethiopia, Germany, Sapienza, Serbia, Belarus, Vancouver, Brazil, and Amazon. And I don't know if that's the company Amazon or maybe the Amazonian rainforest, but in any event, we appreciate all of the great questions and the attendees. And I also want to thank my colleague, Christine Lay, for setting this up and executing it via Sinobiological. Christine, you're always very appreciated. And so with that, I guess we'll sign off, and I wish everybody a good evening or good rest of your afternoon.
Shasha Neiman.
Thank you.